आइए अब हम बात करते हैं टाइप्स ऑफ माइक्रो एरे की। जो हमारा मेन फोकस होता है माइक्रो एरे परफॉर्म करने के लिए, वी बेसिकली वांट टू स्टडी द एक्सप्रेशन ऑफ जीन्स। देयरफॉर द मेन method or mainly microarray is used to analyze the DNA. That is the reason we call it as DNA microarray. इसमें हमारे पास जो मॉलिक्यूल ऑफ इंटरेस्ट है जिसको यूज़ किया जाता है इस मेथड को परफॉर्म करने के लिए, मोस्टली दैट इज cDNA माइक्रोएरेज। cDNA यूज़ करते हैं। उसके अलावा समटाइम्स क्या होता है कि हम cDNA की बजाय शॉर्ट सिंगल स्ट्रैंडेड DNA मॉलिक्यूल्स को भी यूज़ कर सकते हैं टू परफॉर्म दिस मेथड। दैट इज द रीजन इट इज़ कॉल्ड एज ओलिगोन्यूक्लियोटाइड माइक्रोएरे। Besides this, if we want to analyze genomes or different regions of the genomes, then we can perform some other types of microarrays as well, like SNP microarray or BAC microarray. These are different types of microarray which can be performed using DNA as the molecule of interest. लेकिन अगर इसी स्कीम को, इसी प्रिंसिपल को, हाइब्रिडाइजेशन के प्रिंसिपल को सामने रखा जाए और फ्लोरोफोर्स के प्रिंसिपल को सामने रखा जाए, then this is possible that we can prepare the chips using different types of molecules, for example RNA or protein or carbohydrate. Basic theme of this method remains same. Point is that what is your molecule of interest or biological molecule of interest you want to study. Keeping in view that molecule of interest, we can have different further types of microarrays, like for RNA, microRNA population, we can have chips, we can have protein microarrays, tissue microarrays, cellular microarrays, chemical compound microarrays, antibody microarrays, carbohydrate arrays or glycoarrays, and so on. So these are different types of arrays which can be performed using this method, keeping in view the principle, that is the hybridization of the sample with a gene chip or with a microarray that contains large number of spotted molecules of known sequence and of known information. अगर हम बात करें oligonucleotide microarray की, we will be talking about only DNA microarray, और उसमें भी हमारा कंसर्न होगा कि हम बात करें cDNA microarray की, because this is the most practiced method in the laboratories. Oligonucleotide microarray में we have a source of sample. ये source of sample हमारे पास cell culture भी हो सकता है, that can be some tissue as well. So once we do this cell culture, अब ये cell culture अगर देखा जाए, that can be of bacterial nature, which has got the recombinant DNA, or biotechnologically our gene of interest has been inserted in that. We extract the RNA and then we proceed further for reverse transcription, RNA isolation, reverse transcription, prepare the cDNA, then hybridization to microarray, and lastly the scanning of that microarray using the scanner to get the results. The results appear in the form of spots on a computer screen, जो कि यहाँ पे आप देख सकते हैं. So this particular black background slide or part of the slide that reflects the results of a microarray. The spots which are glowing basically shows that the hybridization happened there. cDNA, which was labeled from the source sample, that has been bound here. And if you look at the intensity of the colors, some spots are very bright, some spots are not very bright. That tells us the expression level, how strongly some genes are upregulated and some other genes, they are not upregulated. Then we have cDNA microarray. cDNA microarray is basically used for comparing two different conditions. अगर हम इस पिक्चर पे थोड़ा सा कंसंट्रेट करें, हमारे पास एक कंट्रोल सैंपल है विच इज हेल्दी सेल्स, दूसरा हमारे पास एक्सपेरिमेंटल सैंपल है, दैट कुड बी कैंसरस सेल्स, दैट कुड बी सम सेल्स विच आर अंडर द इफेक्ट ऑफ सम ड्रग. process is similar to extract the RNA, to label the cDNA. Now here we have two different sources of cDNA, of messenger RNA, which are going to give us cDNA. So we use two different cyanine proteins. One is going to label the cDNA in green. The second one is going to label the cDNA in red. Then the next step would be mixing up these two cDNA samples, combining these targets and performing the hybridization. Both these cDNA samples, they are going to hybridize the same chip, and the result would look like this. Some spots will appear red, some spots will appear green, and some will appear yellow. That will indicate that the red spots only get the genes expressed in the normal cells, green spots only get those genes which expressed in cancer cells, and the yellow one expressed in both. Microarray is somehow a complex process which has two parts. One part is the construction of microarray. The second part is the use of microarray to get the results. Normally, the construction of microarray is not the function which is done by a researcher in the laboratory. This is prepared commercially and provided commercially. लेकिन इसकी अगर समरी देखी जाए तो हमारे पास एस्टेब्लिश्ड डीएनए क्लोन्स होते हैं, उनको पीसीआर एम्प्लीफिकेशन के बाद, प्यूरिफिकेशन के बाद रोबोटिक प्रिंटिंग के जरिए ग्लास स्लाइड्स के ऊपर उनका एक मैप बनाया जाता है, एक प्रिंट्स डॉट बाय डॉट प्रिंट किए जाते हैं, और उसका एक इन पैरेलल हमारे पास रिकॉर्ड होता है कि कौन सी रॉ में, कौन से कॉलम में, कौन सा स्पॉट, कौन से जीन का सीडीएनए लेके चलता है. अब ये रोबोटिक माइक्रोएरे इज सप्लाइड to the researcher, and the researcher starts working from sample preparation. Whatever is the sample, the researcher takes the sample and perform the extraction of RNA, cDNA, labeled cDNA, and then using this robotically printed microarray is going to hybridize it. कैसे hybridization होती है, we will talk about it. After hybridization and washing, this microarray is subjected to this scanning machine, and that scanning machine is going to scan for green lights or for red lights, and then superimpose both of these images, and the data is generated, which is later on analyzed. And as the number of genes can be more than 20,000 or up to 20,000, the data generated needs expertise of the bioinformatics experts so that the maximum utilization of the data can be done.